Hello my dear friends, myself is Dr. Avinash Amangi. I am from Gurudev Academy, Dharwad. So we are going to discuss with respect to the chapter called as Plant Growth and Development, which is PUC first year chapter, related to the word synopsis part and concern with respect to the Karnataka Common Entrance Test. So here overallly if you find out, the development of a plant is based on the descendants of what the particularly zygote, and zygote is known as what fertilized egg, which the later period it is going to get what zygote develops into what mature plant through the what growth and differentiation, forming something called as what roots, leaves, branches, flower, fruits and seeds. Then they eventually what they are going to die. So zygote will divide with respect to like these stages. First it forms what two cells. From the what two cells you are going to get it majorly you are going to get it what 8-celled embryo to globular embryo, then heart embryo, then majorly what a tropioid embryo, then it will forms what something called as what plant. So overallly it forms what root, then shoot, and with respect to the root and shoot, the formation of what leaves, branches, flower and fruit and seed even is going to take place. Now we'll see how exactly this particularly plant growth is initiated. Now if you find out the particularly what plant growth, right? So what it means, growth is actually an irreversible permanent increase in the size of an organ or its a parts or an individual cell. It even involves in metabolic processes and that consume something called as what energy. So growth is known as what irreversible change. So it is known to be the permanent increase with respect to the size, shape, and it forms something called as what organ. Even cellular development is going to take place. So during that process, it is going to carry out what metabolic activity, consumes what some form of energy, and form something called as what plantlet, right, or plant body. So overallly if we consider, plant growth continues through the particularly life due to the what presence of what meristems. These meristem cells have the capacity to what divide and self perpetuate, right. Then the growth where the new cells are always added to the what plant body by the activity of what meristems and is called as what open form of what growth. So already we have discussed with respect to the what anatomy of these particularly flowering plants. There we have came across with respect to the what some of the what important form of cells known to be the what meristematic cells. So which has what continuous form of what division capacity. So which will go on continuously divide to form this particularly what plantlet, right. So that type of what growth, what we call it as open form of what growth, right. Then you consider like what how actually the growth can be what seen, or plant growth generally is what indeterminate it can be known as. So primary growth if you consider, it occurs due to the what root apical meristem, shoot apical meristem, and it causes something called as what elongation of the plant along their axis. So this is what even we have studied with respect to the what primary growth, apical meristems majorly which are related with root as well as what shoot, right. Then secondary form of what growth can be seen majorly if you consider primary growth, the intercalary meristems even plays very important role here with respect to the primary growth. Secondary growth majorly it can be seen with respect to the gymnosperm and only with respect to the what dicot. It occurs due to the what lateral meristems, right. Here you can find out what vascular cambium, cork cambium. These causes what increase in the girth of this particularly plant. So girth size can be formed by means of vascular cambium, then or you can consider like what cork cambium, right. The lateral meristems that even increases something called as what girth of this the particularly what the plant, or majorly stem part you can consider, even with respect to the root too. At a cellular level, if you consider, growth occurs due to the what increase in the amount of what protoplasm. So protoplasmic content if it increases, so there you can find out increase in the protoplasm is difficult to measure actually directly. So growth is what measured by means of parameters like what increase in fresh weight, dry weight, length, area, volume and cell number, right. So cell number if we take, the maize root apical meristems can produces what more than 17,500 new cells per hour, right. With respect to the size if you consider, cell size in watermelon may increases in size up to what 3,50,000 times. So that much of what size you are going to find out growth with respect to the what this particularly watermelon, right? Then lengthwise, if you consider growth of what pollen tube, so pollen germination with respect to the stigmatic surface, there you can find out the formation of what length of this particularly pollen tube, right? Then the surface area basis, the leaves or dorsiventral type of what leaves which are surface level, it is going to what increase with respect to the what surface area, right? That what you are going to find out. Now phases of growth, if you consider, that is nothing but what meristematic phase. It occurs in the what meristems at a root apex and the particularly with respect to the shoot apex. The cell in these regions have a rich protoplasm and large nuclei, right? So these are the properties what we have to remember here. Then cell wall are primary, thin, cellulosic with what abundant plasmodesmata, you are going to find out the connection between the cells, you are going to understand, right? Then it occurs mainly like what elongation phase, the elongated form of what growth you can consider. It occurs proximal, just next away from the what the tip, leaving the meristematic phase. Next you are going to find out elongation phase to meristematic zone, right? Then the cell have what increased vacuolation size and new cell wall deposition, you are going to find out. Then maturation phase, it occurs in the what cell further away from the what apex, leaving the apex you can consider more proximal to the what phase of what elongation. Next to the what elongation you are going to find out. The cells attains what maximum size in terms of what wall thickening and even protoplasmic modification. So these are the what property you are going to find out. So diagrammatically whatever they have shown here, the detection of what zone of elongation by the parallel line technique you consider. So parallel line technique can be what used to understand how actually elongated form of what growth can be what seen, right? Then growth rate, which is what even considered to be what arithmetic and as well as what geometrical one, that what we are going to see here. So it is the increased growth per unit time. So arithmetic and geometrical growth we are going to measure with respect to the what a time basis, right? How it is like, first we'll understand arithmetic form of growth, that is nothing but what in this actually the following mitotic cell division, only one daughter cell continues to divide while the other differentiate and matures. On plotting the particularly length of an organ against the particular time, linear curve is actually obtained. So with respect to the height of the plant and even the time, if you consider, we are going to get what a linear form of what curve here, right? Or linear form of what growth can be what seen. So arithmetic form, if you consider one cell divided into two now. Now inside this one will remains what as it is like what in dividing form, and other one what you are finding out, it is going to be what specialized, get differentiate and mature and carry out what different types of what function. That type of what growth what we are going to call it as arithmetic form of what growth. Then arithmetic form of growth can be what measured mathematically, as expressed as mathematically is what LT is equal to L naught plus RT, right? So LT is with respect to the what time what we are going to calculate, length with respect to the time, then length at what 0 time, that is what L naught what we are calling it as, plus R is what the growth rate and elongation per unit time, that what we are going to find out. So based on this we can calculate something called as what arithmetic growth, and by plotting it with respect to the graphically, if you find out, it shows us linear development or linear curve, linearity of what graphical representation can be what seen here. Then geometrical form of growth, you can see here what will happen, two cells whatever are there, they are continuously will be into the what dividing one. So there will not be any what a differentiated form of cells what you are going to find out. Here both the daughter cells continues to what mitotic cell division. In most systems the initial growth rate will be what slow, that what we call it as lag phase, then increases rapidly with the log or exponential phase, and if the nutrient supply is limited the growth slows down and leading to the what stationary phase. Then on plotting the particularly parameters of growth against the particular time, we get what sigmoid curve. So if you want to calculate with respect to the size or weight of this particular organ or with respect to the plant, then when we are calculating, there we have calculated height arithmetically, here we are calculating geometrically weight and size wise increase with respect to the organ you can consider, or overall plant you can consider. So what it gives me, at the first phase I am finding out what log phase, lag phase, which will be what slow, and later period it will go on growing exponentially. right? And or we call it as log phase, right? Now what will happen after a certain period of time, when the nutrition availability is going to be limited, then it is going to form a stationary form of what phase, right? So majorly it shows geometrical form of what growth shows what sigmoid curve, right? Then you consider geometric form of what growth on plotting what parameters of what growth against time, we get what typically sigmoid curve, right? A sigmoid curve is a characteristics of what living organism growing in natural environment, and it is a typical for what cells, tissues and organs of the particularly what plant. This we can apply for all of the organisms actually. Then exponentially, like what growth can be what expressed in the form of what a w1. W1 is equal to W naught into ERT, right? So that is what we are going to get here. So what it means, W1 is final size, W naught is initial size, R is growth rate, T is what time of growth, and E is nothing but base of what natural logarithm. That what you are going to find out. So based on that, geometrical growth can be what measured. Now overall if we find out here, R is actually relative growth rate, what we can consider it is, right? And it is also the measure of the ability of the plant to produce new plant material, that is efficiency index. Hence the final size is W1 depend on to the what initial size, which is nothing but a W naught, right? Then you consider quantitative comparison. If you consider between the particularly growth can also be made two ways, right? One is absolute form of what growth rate, relative form of what growth rate. So measurement, if you consider absolute form of growth is nothing but it's a measurement and comparison of what a total growth per unit time, what we are going to find out. Relative growth is nothing but it's a measurement of a growth with respect to the what a given system per unit time. With respect to the system, common basically what we are going to calculate. So expressed on common basis, example if you consider per unit initial parameter, those are known to be the what relative growth. So that what we should remember when it comes to the what growth rate, right? Then conditions what actually essential for the what overall growth and development, that is what it requires water. Like for cell enlargement, the rigidity of cells help in extension of what growth. Then water provides what medium for the enzymatic activities and needed for the what growth. So overall what we know, all of the species present inside this planet Earth, they depend on to the what water. So water is a medium, it is known to be the what universal solvent to carry out a biochemical reaction. Then oxygen you consider, it helps to what release something called as what metabolic form of what growth, or metabolic form of energy for the particularly growth. Then nutrient consider, like what macro micro elements are needed for the synthesis of what protoplasm and act as a source of what energy even. Then temperature requires, the plant have an optimum temperature condition at which the growth is maximum you are going to see. Then the deviation from the what range could be what determinant towards its survival. Consider living the optimum condition, if there is a deviation like either it increases or decreases, then definitely it is going to affect the growth. Then light and light and gravity, which even affects the certain phases of stages of particularly what growth. So these are the what factors actually essential for the what growth and development, right? Then we will see how actually this differentiation, de-differentiation, redifferentiation or formation of this particularly cell is going to take place. So what is differentiation? Is a process in which the particularly meristematic cells, mainly like what we call it as root apical or shoot apical and cambium, differentiate and mature to perform a specific function. So differentiation is nothing but meristematic cells present with respect to the root and shoot, those are going to divide, and why after dividing they are going to be into perform, they are going to get mature and perform something called as what specific function. That what you are going to see. Diagram you can observe here. In this particularly cell wall and protoplasm undergo structural changes. Capacity of cell division is actually lost here, right? Then loss of what protoplasm you are going to found out with respect to the tracheary element. They also develop a very strong elastic and lignocellulosic secondary cell wall you are going to find out. Carry water to long distance even under the extreme conditions. So these are the what examples what they have given with respect to the what differential form of what cells. Then there is something called as what a de-differentiation. Now this is also what important. Under a certain conditions what you will find out, living differentiated cells regain the what capacity of division. It is going to regain the capacity of division. This is what we call it as dedifferentiation. Like formation of what meristems, again by means of whom interfascicular cambium and core cambium, from what differentiated parenchyma. From differentiated parenchyma, again it can form something called as what core cambium and the particularly interfascicular cambium. So like that, what the meristematic cells again can be formed. The division, cell division capacity can be what regained here. That is what we call it as dedifferentiation. Then there is something called as what redifferentiation. Diagrams you should watch, like how exactly this particularly redifferentiation, dedifferentiation is going to take place. So dedifferentiated cells can what produces cells that again loses the capacity to divide but mature to perform something called as what specific function, and it is called as what redifferentiation, right? Some of the differentiated cells can produces cells that gain the what that gain the lose the capacity of what a divide, right? Whichever got the property known to be what, who lost the property, like what after the losing the property of what cell division, again they are going to be what mature and perform a specific type of function. We call it as what redifferentiation. So plant growth is open actually, that it can be intermediate or determinate, indeterminate or determinate. Then differentiation is also open because cells tissues arising out of the what same meristems have what different structure at what maturity. Then final structure at what maturity of what cell or tissue is also determined by the what location of the particularly cell. Example, if you consider here cell positioned away from the what root apical meristem and differentiate into what root cap cells, while those pushed toward periphery and the particularly mature as what epidermis. Now cells are going to, like what happened, differentiation form, they lost the particularly capacity of what cell division, right? Now again when there is a necessary, then they are going to what regain the capacity of what cell division. That is what we call it as dedifferentiation. Then later period, again whatever this dedifferentiated cells are, those again loses the capacity of what cell division, right, and becomes what specialized in function. That what we call it as actually redifferentiation. And example what they have actually given, the based on the what position basis, like what a formation of epidermis can be what seen. So overall, how actually this plant development, that what we are going to discuss here right now, right? Now with respect to the what plant development aspect, if you consider, it is a process that includes what all changes in what life of an particularly organism from seed germination to senescence, right? Then it is the sum of what growth and differentiation, and it is represented as what follows. So what we know, meristematic cells divides, right, and this division to form what differentiation, we call it as plasmatic growth. Even expansion or elongation is going to take place. Elongated is going to be matured, it is going to get a differentiate, right? Later period, like what it will become mature to go under what senescence, and there will be what a death. So like that of what flow of this particularly plant development you can find out. So diagrammatically, if you consider zygote to 16 cell embryo, then heart shaped embryo to plant to what overall development it will takes place. Again like what adult stage to what again it will form what new species of the particularly plant. So like that what it will go, old plant is going to die, right, after the overall developmental aspect. Right. Majorly, if you consider, there are what some examples of what this particularly shape and size variations can be seen, and the variations based on the what environmental factors, right, of what particularly same plant even you can see. Right. So plants follows what different pathways in a response to the what environment. So with respect to the response, with respect to the environmental conditions, it forms what a different form of what growth, right, or phases of what life to form what a different kinds of what structure. This ability is called as what plasticity. Now with respect to the environmental condition, it shows different pattern of what growth. So that itself what we call it as plasticity, plasticity, right? Here heterophily due to the what phases of what life what we'll see. In cotton and coriander and larkspur, the leaf of what this particularly juvenile plant mature and plant are get what different in shape, right? So larkspur example you can consider, this is the particularly what you can find out, the juvenile, adult or terrestrial habitat basis, there will be a change in the shape of what leaves you are going to find out. The heterophily is due to the what, the particularly environmental condition. So you can find out what a difference in the particularly shape of the leaves produced in air and even with respect to the what, water. So example is what, buttercup. So buttercup different shape with respect to the water you are going to find out, or even with respect to the what other environmental conditions, it is going to show the different form of what growth. That itself what we call it as plasticity. So overall plant development if you consider, which is actually controlled by means of what, different factors. Factors controlling plant development is what, one is intrinsic factor and one is extrinsic factor. Intrinsic factor, what it includes, the intracellular or genetic basis, like what overall development of cell and its genetic constitution, that what actually matter. Then intercellular, like what cell itself is going to produce something called as what, plant growth regulators, right? So these are the intrinsic factors what we are going to see up with respect to the overall development of the plant. Inside the plant these changes or these are actually required for its development. Extrinsic factor, there what matters, the light, temperature, water and oxygen and nutrition even matters over, right? So all if it is supplied, then only like what different types of what intrinsic activity is what actually carried. So nutritional basis you can consider. So overallly we will first understand the particularly what intrinsic form of what growth, how it is with respect to the plant growth regulators, or even what we call them as what, plant growth hormones, or even we call it as what, phytohormones, right? So first we are going to discuss with respect to the what exactly these plant growth regulators, or even we call it as hormones, right? So majorly if you consider plant growth regulators are what, small simple molecule that regulates the particularly what, plant growth, right? So that what you are going to find out, chemically formed molecules or cellularly cell based formed molecules. So based on function, PGPs are two groups, majorly plant growth promoters, which actually promotes the growth of the plant. For growth promoting like activities, cell division and enlargement, tropic growth, pattern formation, flowering, fruiting and seed formation is by means of what, plant growth promoters. So example what they have given, auxin, gibberellin and cytokinin. Some of them are going to what, inhibit the growth of the particularly plant, or when the maximum growth is attended, now the growth should be what, inhibited. So for that purpose inhibitors even required. So for what, growth inhibition activities like dormancy you consider, abscission you consider, respond with respect to the wound, stress and abiotic factors and biotic factors origin, that what we are going to find out. So here examples what they have given, abscisic acid and ethylene, right? Mainly ethylene fit either of what the groups, but it is largely a growth inhibitor, right? Ethylene majorly even it can play important role in as a plant growth promoter too, but majorly it fits up with respect to the what, as a plant growth inhibitor, right? Then you consider auxin. Auxin is being what, the particularly first example of this plant growth regulator, right? So majorly here if you find out, Charles Darwin and his son, mainly Francis Darwin, observed that the particularly coloptile of what, Canara grass, the coloptile, the tip of what, this particularly Canara grass respond to unilateral illumination by growing towards the particularly light source. So it started growing what, this coloptile of this particularly Canara grass started growing towards water light, and it was concluded that the tip of the coloptile caused the bending of the particularly entire coloptile. So entire coloptile is actually got what, bended, as it is started what, moving towards light, and that moment we call it as phototropism with respect to the what, light. Then W went isolated the particularly auxin from this particularly tip of what, the coloptile of what, oat seedlings you can consider, right? Auxin was first isolated from the what, human urine actually, right? W went isolated the auxin which is present with respect to the tip of this particularly what, coloptile, right? But the first isolated in the auxin if you consider, which is of what, from the human urine. Auxins are produced by the what, the particularly growing apex, mainly like what, stems and roots, forms where they migrate to the what regions of their actions. Majorly if you consider auxin, you can find out with respect to the apical meristem as well as what the particularly apical root meristem and apical shoot meristems with respect to the tip. And even whenever there is an action required of it is going to move towards that particularly what action, right? Then types of auxin, if you consider, naturally formed auxins are what? Indole 3-acetic acid. You should remember which one is the natural and which one is the artificial or synthetic one. So indole 3-acetic acid and even indole butyric acid, IBA, IAA, they are what? Isolated from the particularly plant. Synthetic form of auxin, if you consider NAA, that is naphthalene acetic acid, and 2,4-D, that is nothing but a 2,4-dichlorophenoxyacetic acid. So these are the what? Two natural and two majorly like what? Synthetic type of what? Auxins. That what you should remember here. Now, what exactly the function of this particularly auxin is? Auxin is to what? It initiate the rooting in the stem cuttings of the particularly plant propagation, what you are going to find out. It promotes the flowering, majorly like example is what with respect to the pineapple, and to prevent the fruit and leaf and fruit drop at the what early stage, that even can be prevented by means of auxin. Promote something called as what? Abscission of what? Older leaves, fruits you consider. Induces parthenocarpy, example what? Tomato you consider. They are used as what? Herbicide even, like 2,4-D is used to kill the something called as what? Dicot weed, right? It does not affect with respect to the monocot plant. So it is used to prepare the weed free lawns you consider. Control mainly like what? Controls the xylem differentiation and even the cell division can be what? Controlled by means of what? Auxin. So these are the what? The particularly examples of what auxin you can find out. Then majorly auxin, if you find out what it is going to do in higher plants, the growing apical bud inhibits the growth of what? Lateral bud. So if the particularly apical bud is growing, with respect to that what it is do? It inhibits the growth of what? Lateral bud, and it is known as what? Apical dominance. It is known as what? Apical dominance. Then what they are going to do is removal of what? Shoot tip, mainly decapitation, what we call it as, result into the what? The growth of what? Lateral bud, and it is applied in with respect to the what? Tea plantation and hedge making. Majorly what they will do, they will remove this particularly what? Apical bud or apical majorly like what? Apical tip of this, and then the lateral like what? Bud is going to grow up. So that what you are going to find out. So like this we can overcome something called as what? The particularly apical dominance. Then there is one more, that is what? The gibberellin, right? So that is nothing but these are the acidic PGPR. Like E. Kurosawa treated the what? Sterile filtrate of Gibberella fusiculae, which is a fungi that actually causes a disease known as what? Bakane disease, or the foolish seedling with respect to the rice, right? So this is particularly what? The stunted growth what we are seeing, this is due to the what? The particularly Bakane disease only what happened to the healthy rice seedling you consider. As a result, it showed the particularly symptoms of what? Bakane disease. Later, the active substances were identified as what? Gibberellic acid. So name is what? The fungi known to be Gibberella fusiculae. From there, they have found out what? The gibberellin or gibberellic acid. So only they have given the name as what? Gibberellic acid. So there are what? More than 100 gibberellin you are going to find out. Like GA1, GA2, GA3, and like blah blah what you are going to find out, right? Then with respect to the what? Fungi and higher plants you consider, there you will find out the presence of this particular gibberellic acid. Gibberellic acid is one of the first discovered and most extensively studied gibberellin. Which one is? GA3, or even we call it as terpensis, right? So terpenes is known to be the what? The gibberellic acid. GA3, which is what? First discovered and most extensively studied gibberellin, right? Then function, if you consider, they causes what? Increase in the length of axis. So they are used to increase the length of what? The grape stalks. So grape elongation form of what? Growth can be initiated by means of gibberellin. To elongate and improve the shape of the fruit, such as what? Apple you consider, for their shape and size improvement required gibberellin. They delay the senescence actually. Then so the fruit can be what? Left on the tree to extend the particularly market period. Even senescence means the lifespan is going to like what a delay over here with respect to the using such kind of what gibberellic acids. Then GA3 is used to speed up what malting process with respect to the what beveraging industry. In beveraging industry, for the malting or to carry out a fermentation procedure, there also they use this particularly GA3 terpenes, what we call it as. Again, functions, if you consider, sugarcane stores, mainly what sugarcane stores sugar in the stem. Spraying the particularly sugarcane crop with the gibberellin increases the length of this particularly stem. Then it increases the yield as much as what 20 tons per acre, like that you are going to find out. Spraying the juvenile conifers with the GA3 hastens maturity, right? It leads to the what early seed production even. Then for bolting, you consider internode elongation just prior to the what flowering you can find out in beet, cabbages and many plants what you are going to find out, the rosette habitat. So bolting procedure, for that even gibberellic acid can be what used, right? Then overally like next is what cytokinin, that is what the mainly F Scoos and the co-worker observed that from the particularly internode segment of the tobacco stem, the particularly callus started growing. And what is this callus? Callus is nothing but it's an undifferentiated mass of cell. So because of this invention of what cytokinin, majorly what you are finding out over is this Scoos developed something called as what the tissue cultured media, which is known to be the what MS media. Morarsis Coos media, what we call it as. Then mainly callus is nothing but what mass of what undifferentiated cell, which actually proliferate only if the nutrient medium was supplied with extract of what vascular tissues, yeast extract and coconut milk or DNA. So this callus, which is actually grown up with respect to the tobacco plant, if it is provided with respect to the nutrition, then definitely it is going to differentiate as what plant. So that what they have observed, right? And the Scoos and Miller later identified as what crystallized active substance which was actually present and termed as what first as a kinetin. Then kinetin, later period cytokinins were discovered as what kinetin. First they discovered it as what kinetin, right? Here you consider what is that kinetin, N6 perfural amino purine it is, right, which is an adenine derivative, which is known to be what an adenine derivative. From the what autoclaved herring sperm you consider the particular DNA. Then kinetic does not occur what naturally with respect to the what plant. You are not going to find out naturally being present with respect to the what plant, right? So that you can see, right? Then cytokinin mainly what zeatin from the what corn kernels or coconut milk is the what natural substances with the cytokinin like activities. Then there are what some synthetic compounds with the what cell division promoting the particularly activity. The natural cytokinins are what synthesized in a region of what rapid cell division. Then root apex or shoot bud apex or with respect to the young fruits. So there this particularly kinetin and even what zeatin can be what used for the what development of particularly plant. Then function, if you consider with respect to the cytokinin, they play a role in cytokinesis, right? Then majorly cytokinesis is cytoplasmic or cellular division you are going to find out. They help in to what produces neve leaves, chloroplast in the particular leaves, lateral shoot growth and advantageous shoot formation can be found out. They help in what overcome the apical dominance. Then again they promote the nutrient mobilization which help in the what delay of what leaf senescence. So these are the what functions what actually we have to remember. So apical dominance even can be overcome by means of what cytokinin. Then there is one more hormone ethylene which is actually known to be the what inhibitor, but even it shows some of the what growth promoting, right, hormones. So cousins like confirmed the particularly the release of what volatile substance from the what ripened oranges that hasten the ripening of this particularly stored banana. So with respect to the ripened oranges, the volatile substance was actually released and which actually carried out what the ripening of what even banana, right? Later this substances was identified as what ethylene, right? Then ethylene is a what simple gaseous PGPR and it synthesized in a large amount by a tissues undergoing senescence and ripening procedure. So it will make the particular tissues or the cells to undergo what the senescence, and which forms what something called as what ripening of this particularly fruit. So fruit ripening, they you will find out this gaseous element that is nothing but known to be ethylene. Now function, if you consider, it influences the horizontal growth of what seedling, swelling of what axis and apical hook formation in the what dicot seedling. It promotes the senescence and the abscission of particularly plant organs, especially of what leaves and flowers. Right. It promotes the fruit ripening and it enhances the respiration rate during the what the particularly fruit ripening. This is called as what respiratory climatic. Right. Then again it breaks the what seed and bud dormancy. It initiates something called as what germination with respect to the peanut and sprouting, even potato tuber you can consider. So overall these are the what some of the functions of what this ethylene you can find out. Right. So somehow if you want to read these particularly aspects, whenever you are reading, you just pause one's video and you can find out the particularly even functions of these hormones, right? Majorly if you consider, even this promotes what rapid internode petiole elongation, there also it will help. This ethylene has both the positive as well as what a negative role. So only you can find out majorly here with respect to the plant. In a deep water rice plants, you will find out internode and petiole elongation. It help in leaves upper parts of the particular shoot to remain above the water, and again promotes the what root growth or root hair formation. This increases the absorption surfaces, right? Then used to initiate the flowering and forward synchronizing the fruit set with respect to the pineapple. It also induces the flowering of what mango, right? Then again it is widely used in what the agricultural purposes. So overall even some of the images what we have shown over here, right? So that you can find out, right? So ethylene, if you consider, like most widely used source of ethylene is what ethepon, right? Which is ethepon is an aqueous solution and is readily absorbed and transported within the particularly plant and releases what ethylene slowly. When you transfer ethepon liquid form, then later period it forms something called as what ethylene, which is gaseous element. Right. Ethepon hastens what the particularly fruit ripening with respect to the tomato and apples, and what accelerate the abscission even in what flowers and fruits, that is what thinning of what cotton, cherry, walnut you can consider. It promotes the female flower in the particularly cucumber, thereby increases the particularly what even yield. So that what you can find out with respect to the ethepon. Then there is one more inhibitor which is nothing but what abscisic acid, even what we call it as ABA. Right. So during mid 1960s, it was reported that the three kinds of what inhibitors, inhibitor B, abscission 2, and even dormine, what we call it as. So they were the chemically identical and now known as what abscisic acid. So all these three were discovered in 1960s, but what they found out chemically they are what identical. So only they are all together called as what abscisic acid. ABA is derived from what carotenoids. It is actually derived from the carotenoid with respect to the what the leaf you can consider. It regulates the abscission and what dormancy, that even you can find out. So overall functions of what this particularly abscisic acid, if you consider, it act as what the particularly inhibitor of what plant growth and metabolism. It inhibits the plant growth, even metabolic activity. It inhibits the seed germination. Then it stimulates the what closure of what this stomata in epidermis. So closing of stomata can be formed by means of what abscisic acid. It increases the tolerance plant to the what various kind of what stress. Therefore it is also called as what stress hormone. Now which is the plant stress hormone? That is what abscisic acid, because it increases the plant or make the plant to be resist with respect to the what stressful conditions, right? It has an even important role in seed development, maturation, dormancy. Seed dormancy by what ABA helps to what withstand the particular desiccation and other factors which are what unfavorable for growth. So like how the particularly plant growth are required, plant growth regulators are required, like that only majorly plant growth inhibitors are also required. So majorly interactions of PGPR, if you consider, PGPR play individualistic or synergetic role. such roles may be what complementary you consider, or even antagonistic type you are going to find out. So PGPR interact to affect the dormancy in the seed buds, abscission, flowering, senescence, vernalization, apical dominance, seed germination, plant movement, etc. And in most cases ABA act as what then a antagonistic to the what GA, that is what gibberellic acid. So that is what even is what important. So overally, like how actually plant growth hormones are required, mainly like sorry plant growth regulators are required for the overall development of the plant, like that only inhibitors are even required. So somehow with respect to the what being in stressful conditions environmentally, or even different factors which are going to affect the growth and development, their inhibitors are even to be required. So interaction of this particularly PGPR is what required. So majorly if you consider overall development of the plant can be even affected by these particularly extrinsic factors, correct. So extrinsic factors where the light, temperature plays very important role, even different form of water content or whatever, these things like what overall development of the plants are required. So extrinsic form and intrinsic form, temperature exposure, and even what the nutrition availability of the what plant, light exposure even you can consider, that forms something called as what overall growth of the particularly what plant. So this is all about this particularly chapter, that is plant growth and development. So thank you so much. Please do subscribe our channel and share and like, and even prepare well for this particularly what KCET examination. So all the best. Thank you.